Welcome everyone to the CCAIM seminar series. Welcome to 2024. We have a really nice program planned for you throughout the year with speakers coming in through the month. So keep an eye out on our website. And I'm very happy to start this year with one of our newest members of our associate faculty in the center. And I leave it to our director, Professor Andres Floto, to introduce today's speaker to you, and looking forward to hear from him. Thank you.
Thanks, Andreas. Hi, everyone. Welcome to this edition of the seminar series from CCAIM. My great pleasure to introduce Mo, who, as Andreas mentioned, recently joined the Centre as Associate Faculty and recently joined the Sanger as a group leader. So Mo, without further ado, over to you. Really excited by the talk.
Thank you very much, Andreas. Okay, let me just see if I can share my screen. Yeah, thank you very much for having me. So today I'm going to talk about a series of work across, I think I made it kind of broad, and also for the broad audience, I left out some of the method part, the math. If you have further question about the math, you can look at the papers or also reach out to me. I can share that. But yeah, so in addition to my academic positions, I'm also director of machine learning research in relation to therapeutics in London. And my research is an intersection of machine learning and biology, cellular biology. And most of the research that I show you today is using high resolution data from single cell biology. So for those of you who are not familiar with single cell biology now we have the ability and the technology to get a sample from a patient, and then for that sample, basically isolate every individual cell, and then capture different molecular information from this specific cell. So that could be gene expression, how the gene expression actually changed, and then chromatin accessibility, protein abundance, and also like location of the cell in a tissue. And obviously, then what would be interesting is what happens to these cells or individual cells after a certain perturbation. Perturbation could be disease, or what happens to the cell if, like, internal cell state across these different modalities if we apply certain drug or genetic perturbation. So yeah, so as I said, so today I'll talk about how single cell biology, so high resolution biology combined with machine learning, can help us to understand health and disease across different scales. For example, can we look at the similarity across different patients based on individual cells of each patient? Can we zoom in into one patient and understand the heterogeneity across different cell types? And even model how one individual cell actually in that specific patient responds to a certain treatment. And finally, if I have time, I also talk about how we can actually map the cell types in the tissue and understand how they actually are organized in the tissue, how they communicate to each other, which is also a big part of understanding tissue biology. Okay, so I'm also part of like a larger consortium called Human Cell Atlas. And the goal in Human Cell Atlas is to basically create a map of human body similar to a Google map, so such that we know the exact location of every single cell in our body. And then we have different technologies to achieve this. So we have single cell technologies to capture different type of information, gene expression, protein abundance, the location of the cells, and so on and so forth. So ultimately, we would like to turn this into a digital entity. So such that for every organ atlas, and we call these atlases because they are comprised of many cells for that specific organ or tissue, and tie it to a digital version of that tissue, which could be a neural network or large scale transformer model that is trained on many data sets, and that could be used as a basis of further downstream analysis or to model changes in the tissue. And when the word atlas comes in, it basically implies a huge resource, and that means that that resource can be reused for downstream analysis. And that's a pipeline that I think we kind of pioneered a few years ago, that can we actually use these reference atlases to analyze query data sets? Because analyzing single cell data sets is actually quite time consuming, because you have to take those data sets, pre-process them, and put it into some sort of clustering algorithm, identify those clusters, and then perform some like expert annotation of each cluster, and then try to iterate and optimize this process. But reference-based analysis has been used in also other domains. So it's not a very novel concept, but we, especially in biology and in genomics, we use it day by day. So we have a sequence a reference genome for human or for other species, and then we try to reuse this sequence map to map new data and then understand the similarities and differences between the query sequence and the reference sequence. So the idea is, can we do the similar analysis now for cellular biology? Meaning that we create a huge atlas, and as similar to reference assembly, there are different algorithms to create these reference atlases. So they are like reference building tools and methods. And then we have a different set of tools that are reference mapping. So given a pre-trained model, given an assembled reference, can we give it to this reference mapping algorithms and a query data set to map this query data set on the top of a reference atlas? So we're talking about aligning a new data set coming from completely different technology or different lab to a reference data. So the reference atlas could be made from like multiple modalities, as I kind of outlined. And so this was the idea of kind of like now having similar type of paradigm for single cell biology, such that we create these pre-trained models. And these pre-trained models, as I said, there are like many of those out there, and so I'll just keep it generic at this point. And so the only assumption is there is sort of like identifier for a data set, meaning that you have the gene expression, or it could be multiple modalities, so that's Xi. And then you also have a label called Si, and this Si is an identifier that this cell belongs to study I or J, etc. And these models are trained to regress the effect of the effect of the study across, or technical variability across different studies. Because as you can see, we have like N studies, so that come from like different labs, different universities, and different experiments. So we need to correct for the batch effects, or as in more like computer science literature kind of domain effect. And in this case, it's a very simple generative model that actually marginalize the effect of the batch and then give you a latent space Z, which is free of these domain effects. So the question is, can we actually create a model zoo? So having this pre-trained model, push them into a model zoo, and then perform transfer learning and reuse them for downstream analysis. And in our case, what we're interested in is like, if we have a healthy atlas, can we map disease data and then contextualize the disease into the healthy and identify which cell states and will cell type actually change compared to the healthy reference, or transfer information across species, or if you have a certain atlas built for a certain organ, you can map another organ and perform cross-organ analysis. And in the simplest case, you map a new data set from the same organ and tissue to analyze that data set. So this was the idea behind the study, which we call SCArches. So we introduced a kind of low cost and also well-designed fine-tuning strategy that we take a bunch of these generative models and then we add and inject new nodes, such that these new nodes could separate between the biological variability and the technical variability. So these nodes are only attached to the part of the network that actually models the dataset variability. So these dataset identifiers. And then we only fine-tune these labels. So that assumes that you assume there is the biological variability and the technical variability they kind of like orthogonal, because we freeze the whole network. That means that we want to keep the coordinates of the atlas, so the reference coordinates, fixed, and then we only fine-tune these specific weights. So in the paper, we provide further evidence and also lots of experiments to show that why this work. And but the nice part of this is actually order invariant. So meaning that if you have a pre-trained model, you can iteratively add new data sets and basically update this network with new data sets. And very similar analogy to also lifelong learning, and then you can actually update these and then push these adapters and share it with the collaborators. So this was published 2 or 3 years ago, and this was a basis of kind of modern cell atlasing, that everyone now kind of use these models combined with some sort of reference mapping algorithm to create these large scale atlases and then reuse them for downstream analysis. One example here is a recently published atlas to create the first human long cell atlas, HLCA, led by Lila Sekema and then Malte Luken, and also collaboration with, I think, many other universities that they kind of made the first core reference across 500,000 cells across many studies, and they use SCArches to extend this atlas along 2 years, to extend it to 2.2 million cells across 445 different patients, 46 data sets, and 600 plus samples. And now they even further updated that you have 30 different diseases. So this can actually be a basis of analysis of every human lung disease, because you can easily contextualize them in this atlas. So what is the usage of this type of reference mapping? And so nowadays people actually use it to, as I said, automate the analysis. So meaning that you project the data and then you transfer cell type annotation from the reference to the query, assuming that you corrected for the batch effect. But the most, I think, interesting usage here is to map a new data set and use this reference map to identify whether you have novel structure or novel cell states in the query not present in the reference. So what they did and what they showed in this manuscript is, if you use this model and map a new long data set, so you see these are the UMAP representation of the latent space of this model. So the gray is the reference and the blue is this new map query data set. And then they transferred cell type annotation, and they used a very simple technique based on a KNN classifier trained on this aligned latent space of the reference and the query to transfer cell type annotation from the reference to the query. And using the kind of the uncertainty of this classifier, so how sure is the model is about assigning a certain label to this query, to each query data cell, to identify the unseen structures and unseen cells in the query. And so what you see here is the distribution of these uncertainty values projected into the 2D representation, plus the correlation or the relationship between this uncertainty value and how well these cells were annotated. So what they looked at were the cells that they had the higher uncertainty, they were actually the ones that were absent in the reference data set. And then there are after this paper, there are multiple other papers that they kind of use the same paradigm, but to also project the disease and understand, are there also novel phenotypes or novel cell state that would arise in disease. Another use case is which I quite like is to compare between the in vivo and in vitro samples. So in this case, this is not my publication, but I think a very good example of how different, how kind of these large scale modeling and reference mapping could help to analyze the data, In this case, they use this model to create a time-resolved atlas, single nuke resolution atlas across human prefrontal cortex development. So they have different samples from a very early fetal stage toward adultness, and then what they do is they query this reference atlas with different type of queries. For example, they map glioblastoma, a very lethal tumor in brain, to map it to this healthy reference and identify the gene programs that happen to be in glioblastoma, but also in a very early stages of development to be reactivated in the disease. What they also did was mapping different gestational stages to this map, and then annotating each cell because they have a very nice time result data and age for each single cell to annotate the real age actually in this other data set, but also see the differences across the development. But what I like most is you can actually project in vitro data. So the brain organoids to this in vivo reference and see how well you can capture the true underlying mechanism in brain actually in the lab. So see if there are, there could be some cell states and cell type that happen to be in vivo, but not in the organoid model, and use this for protocol optimization. And this is for the age prediction task, so they can kind of formulate this type of task. But the question is, can we go beyond querying cells and look at these models as some sort of database, as you kind of I think every one of you use ChatGPT, so you can just have more interpretable queries. So can we use these large atlases to ask which gene programs are changed after disease, or what is the effect of a certain drug? Which cell-cell communication are disrupted when you have between in health and disease. So when you have disease, and there is a challenge here, because most of these models are based on highly nonlinear deep networks, where you most of the time actually lose the interpretability, identifiability of the source of variations. So that's not really satisfying. If you show this to a clinical or a clinician or a cell biologist, they're like, okay, but what are these axes of variation? What do you represent? So the question is, can we go from this to a regime where you actually have an annotation for each latent dimension, so each axis of variation of the neural network? So you can see, for example, there is an axis of variation for T cell differentiation. So whatever you see in this axis of variation could be map of the dynamical changes of gene expression along the T cell trajectory. And the same thing for the inflammation. So this was the idea that we kind of wanted to make these kind of queries possible and then make these models interpretable. So we came up with a mechanism that we call it, or a model that we call it XPMAP. And the idea is to use the domain knowledge. So the domain knowledge about the gene programs, which are actually annotated, and it's kind of common language. So if you show these gene programs, for example, about interferon signaling to a cell biologist, they're like, yeah, okay, I know this. And so the idea is we want to create a matrix, and each row in this matrix is actually a gene program, and each column is a gene. So if you have a gene as part of this gene program, you just put a 1 there, and then so it's kind of like a binary matrix. So the idea is, or the challenge is, how to put this domain knowledge that we have from literature, from experts, from databases into these neural networks, such that each latent dimension of this neural network actually captures the variability with respect to that gene program. So you can disentangle the effect of different gene programs. And it turns out it's not super easy. So you can't just apply that, because it's a very sparse matrix. Then, suddenly the optimization algorithm breaks because of the sparsity. And then what the idea that we came up with was kind of linearize the decoder and the encoder-decoder architecture, and put a kind of hard mask on the top of each gene program, so each latent dimension, such that each gene program or each latent dimension can only reconstruct a certain set of genes. And that brings certain limitations because you always, there's like a lot of noise in this type of domain knowledge. So there might be genes that are not, relevant for your data set. They're kind of like, they shouldn't be in that gene program. So we need to have a mechanism to prune these gene programs, meaning that you need to add or remove different genes to this gene program based on the data. So you have to learn this. And ideally, none of these gene programs are relevant for your data set. So you also need a certain mechanism to not just prune the features, so the genes in the gene program, but also prune different gene programs. And finally, you would like to go beyond domain knowledge. So use these models to actually learn new gene programs, because there could be certain, or like a new disease that, the gene program regulating the disease or affected by this disease is not part of the domain knowledge. So we also design a mechanism to actually allow the model to learn novel gene programs. And then these gene programs should be novel enough compared to the reference set of gene programs. And then we came up kind of like with a nice formulation using Hilbert-Smith criteria to kind of enforce these models to be, or these learned gene programs to be independent to the previous ones. So I'll show you one example. This is actually applying this model to create a healthy PBMC atlas. So these are all blood cells, and mapping a new data set that contained both control, so not perturbed cell, but also the cells that were perturbed with interferon beta. So interferon beta is a very strong stimuli that causes global shift and very strong shift in the gene expression compared to the healthy or like control condition, but also has specific changes that only certain cell type happen to have. So once you map this using the same mechanism that I explained in the SCArches paper, the question is, what are the main mechanisms, or what are the main gene programs that cause these interferon stimulated cells to be separated from the healthy cells? So the model is actually a probabilistic model. So you can come up with a closed form by sampling across different gene programs that allows you to do hypothesis testing. So you can ask, what are the main drivers that separate the red blob from the blue blob? And then you come up with these like interpretable terms, i.e. the latent dimensions that separate between the control and interferon. And obviously, just like here, as we expect, they're mostly dominated by interferon-related gene programs. And if you project the data now into this latent dimension, in these top two latent dimensions, then you can see that the most variability actually belongs to the interferon signaling. So all the red cells, they kind of separated along these two axes from the blue cells. So you lose the cell type variability, but here you can actually perform the same analysis. So you perform the same test to see, oh, okay, what are the most important gene program for B cells? And if you combine this with a perturbation gene programs, what we did was basically looking at the mechanism of interferon signaling to see how this perturbation actually affects different cell type. Is there any cell type specific response? And it turns out indeed, so this is also well known in the field, but what we also explored here is what we capture here that was not reported before is actually change of metabolism that could be captured using this model, and that is only specific to myeloid cells. And so I don't go into the deep detail. This is one application with how you can use this. But what is nice here is actually the knowledge that you put into this model increases the performance here. So we compared this model across with like existing work, and what we observed was if we basically benchmark it against like models that are not regularized to using this domain knowledge, and we observed that you can actually outperform these models, although we have a linear decoder. And we have a nice proof in the paper that actually we show this domain knowledge actually reduces the amortization gap. Is a very big problem and mostly in variational autoencoders and variational inference because of the amortization process. And we're basically on par with a model which is not amortized. So we basically
have the model scale with the number of cells. And this all comes from the mathematical background of this model, is a formulation of the model. But yeah, in addition to type of like, yeah, empirical result, we also have a nice proof, so you can check this out in the paper. So yeah, I'll move to the second part of the talk. So I'll talk about, now I talked about single cells, cell types, but the question is, how can you scale these models and also look at the patient variability and the donor variability in addition to the cell variability? So you would like to have two different latent spaces here, one to encode the variability across population and one that encodes the variability in cells, and then you would like to link those. And this allows you to kind of like ask questions like, where is my population? What is similar, different across different samples? And then later in the later stage, we can like extend these models to explicitly ask the model what are the certain or important cells without actually manual analysis for a certain disease. So this is a recent work from us called SCPoly. So it's a conditional generative model that received gene expression and also the cell type. So it's kind of like semi-supervised model and also the cell type labels. And then we learned two different embeddings. So one for patients and donors and one for cell types. So I'll skip the modeling part, but it's kind of combining the generative model with the idea of like meta learning. So you actually do meta learning across different cell types, and then allow to kind of like transfer to new tasks, which are different node donors or different cell types in the test. So yeah, I'll show you one example that we actually recreated the human long cell atlas using this model. So what you could see is like this map after the integration is called is colored by cell type and study. But you also have the donor embedding or the sample embedding at the bottom. So each dot is actually a sample, so you don't see cells anymore. And what you could see is you have different type of variation across different samples coming from either the study differences or also the subject type, whether you had certain feature in the donors. And these are all because this is based on prototype learning. You could also project data. So all of the tasks that I kind of introduced using the simple K-NN classifier, you can actually have this type of like uncertainty quantification using a very simple distance-based label transfer. And it actually performs better compared to like to kind of like pipeline approach. But what is nice is now you can actually query these atlases at both levels. So you can project the data, annotate each cell for each patient in terms of like the cell type labels, but also annotate the disease severity or like the class of the disease or like stages of the disease for that specific patient. So what you see here is you created an atlas across 500,000 cells, 270 different samples in different across different COVID stages. And then we held out a bunch of samples of donors that were never seen during the training. We projected these donors using this model, and then we annotated both cell types, but also the disease severity, as you can see on the right. So this is basically the sample space, the patient space. And you can see like the query patients with the ground truth phenotypes are well aligned with the reference. And this turns out to be better than like some other existing methods. So now the question is, this gives you high resolution information about the patient and also high resolution information about the cell type. But if you want to do and identify the novel cell state, because you don't have inherently, you don't know what or which cells are exactly perturbed by disease in each patient. You know the disease sample and the label, but you don't know exact cells that were affected. So the idea is, can we now automatically perform this analysis? So not just annotate the patient to say, oh, this is certain stage of cancer or this is certain tumor type, but also individual cells that actually explain that prediction. So this is also very similar to, in machine learning, multi-instance learning. where we have different bags of cells. So you have like bag of cell, which is coming from a healthy patient, a bag of cells coming from a diseased patient. And the idea is, and also commonly used in whole slide imaging and pathology, where you want to annotate different tumor types. So in our case, we assume we have clinical data and also omic data across patients. And then we design a generative model to actually integrate across different modalities. So I'll not talk about this model. But then once this model is trained, we would like to query a new patient, so project a new patient and annotate individual cells in that patient, but an overall overly basically annotating the whole patient, but identify the cells in each patient responsible for that specific prediction of disease severity. So what you see here is this idea could be applied on any pre-trained model, not that multimodal structure that I kind of showed. So here we showcase an example that a large foundation model is actually trained on like 60 million cells. And then we take these pre-trained embedding, and now we feed it into this multi-instance classifier that we modified to be applied on single cell data. And we held out a bunch of samples. So patients from IPF that they were actually diagnosed with IPF, and also we also had samples from healthy, and showed this kind of like multi-omic data integration plus multi-instance learning actually beats other type of classification algorithm, based both on gene expression, but also compositional differences, because you could still classify a patient by counting how many cell types are, or how many cells from each cell type is actually present, because you have a depletion of certain cell types after disease, or like or emergence of certain, or changes of composition in different cell types. And this is actually better than that. But if you want to identify which substates are actually present in disease and not in the healthy, this model because it's based on attention mechanism, it could highlight where in each patient and each cell type you have cells that were actually identified, or like had the model actually highly attended on those. And here you'll see the attention map learned by this model compared with like some domain, some expert annotated pro-fibrotic score for each cell type. And they kind of like nicely align to each other. And it's kind of significant. So it kind of automatically not just annotate each patient, but also highlight individual cells for each patient that could be further used, for example, for like target identification or drug discovery. But also extend the domain knowledge that we have about pro-fibrotic score gene programs. So you can take this model, look at these cells, and then identify which genes actually changes in healthy and disease. Right? So that was the second part, but now I'm going to move toward actually modeling the changes and predicting the changes in single cell. So the underlying question is, can we predict the response of each individual cell to an unseen stimuli? That could be an unseen drug or unseen genetic perturbation or other type of, for example, disease. So why is it useful? Because this can give you a high resolution information about which cells need to be, or basically, it's kind of used in so I don't want to go deep here, but like this is very much used in a paradigm in drug discovery called phenotypic drug discovery, because in phenotypic drug discovery, you usually don't have the exact source of disease, so you don't know the target, so you can't come up with a certain drug to just hit that target, but you know the phenotype differences between the healthy and disease. And now you want to do some large-scale screening to see whether you can find a drug or a perturbation that would push cells from the disease state toward the healthy state. But imagine that you have access to all the space of small molecules. So, and then even their combinations, which is commonly used in cancer combinatorial therapy. And now you want to have a high resolution readout like single cell. So that's not actually possible, because that's going to be very expensive and you can't really do that. So that was like a topic of like my PhD and also like after that, to come up with algorithms that can actually predict the response. So you can actually start with a very sparse experiment and then predict the response at a cellular level to unseen perturbations. And this could be actually used to design new experiments. So this is also very much linked to experiment design to narrow down the hypothesis space for identifying or discovering certain drug. Okay, so so one of the latest work that we had to solve this problem is called CPA. So this was a work that I've done during my time at the Facebook AI, so collaboration with David Lopez Paz, that in this work, we wanted to disentangle the effect of perturbations or other type of variations, such as cell type variation, patient variation, and basically go back to the basal state of the cell without having any of these variations. And then do it in a kind of like interpretable way. So for this type of task, we actually decompose the latent space. So that's where the name actually compositional comes from. And then try to decompose all of these effects and linearly learn them in a nonlinear latent space to reconstruct the gene expression. So the way that it works is when you have cell A perturbed with 2 drugs, the model learns to disentangle the drug effect and also the cell type effect and infer this so-called basal state, and then put them together and reconstruct the gene expression. So at the test time, what you have is you can actually perform counterfactual questions. So you have cell A perturbed with these 2 drugs, but now you also have cell B perturbed with other set of drugs, but you want to know what will happen to cell A if it was perturbed with those 3 drugs, and then predict that. So we applied this idea. So we developed a method, and then we applied this method because you actually learn individual representation, not just for cell, but also for perturbations. We applied this data set to a perturbational data set, but this time not drug, but genetic perturbations. So this is actually CRISPR-A. So they activate certain transcription factors that could push cells toward in the genetic interaction manifold to a certain cell state. What you see here is actually individual perturbation embeddings that the model learned for either single perturbation or combinatorial perturbations. And the map could be interpreted in a way that the perturbations that are embedded close to each other, those are actually the ones that have like similar impact on the gene expression. And we actually annotated those, and you can see that the actually different gene programs. So you can map the perturbation to the function and see which perturbation could actually, for example, arrest the cell in a certain cell cycle or induce certain lineage. And then we show that you can actually predict unseen perturbations at cellular level. So in this case, CBL plus CNN1 was not seen during the training. So we feed in control cells after training, and then we ask the model to in silico apply this perturbation and predict the response. And we showed during the paper, it goes beyond just linear effects. So you can actually predict synergistic interaction and epistatic interaction. There are like certain limitations that the model needs to see similar examples, not from this perturbation, but maybe from other perturbations. So, for example, if you have neumorphic interactions, there might be cases that actually the model fails. So, and also we apply this model on cancer data set. So at single cell level. So in this case, we applied on a highly multiplexed data set across 3 different cancer cell lines, perturbed with 188 drugs across different dosages. So in total, you have like 4,800 conditions. And the idea was, and this is an extension of this paper called ChemCBA published in last year in NeurIPS, that because we don't have much data at single cell resolution, can we actually transfer information from like other sources of variation, such as bulk, which is the different resolution? You don't have single cell resolution, but there you have a lot of perturbation screen data sets. And so we came up with a transfer learning mechanism to actually transfer information across these 2 different resolutions, but also give the model ability to go beyond the perturbation that it has seen during the training, and then predict unseen drugs. So the takeaway for this model was like, the transfer learning from bulk to single cell can actually help to predict the unseen drug. So this is like a zero-shot example where you haven't seen that drug and you want to predict what if you apply this drug on the cell line. We followed this up to see if we can actually use this perturbation embedding to narrow down the hypothesis space and then come up with the new drug combinations that would give us a certain phenotype. So we use this perturbation map that we have from the first pre-trained model to identify the drugs that were most effective and select 32 of like those drugs and their combination and generate another follow-up, like a validation single cell data set, but use this previous experiment to, you know, narrow down the hypothesis space and come up with a drug list. And then show the model that actually, that this model actually generalizes not for single drugs, but also combination of the drugs for unseen drugs in the training. Okay, so I'll skip this part, but to go to something else that, yeah, so now we're extending these models to kind of like combine the idea of like counterfactual prediction from the causal perspective to the disentanglement, because that's what we actually did in the CPA paper. But now we actually theoretically going deeper and then to see whether there could be identifiability conditions, and then we came up with like kind of like nice encoder decoder architecture. And the take-home message is, yeah, you can do you can have like multiple latent spaces. And this is not yet published, but what we found was like really interesting. If you kind of like derive the math, and in the end, you'll kind of end up with having two different losses. So one that actually causality for like counterfactual prediction, one for the disentanglement. And then we try to capture what will happen. Are they actually feeding in each other? So meaning that if you have a better disentanglement, you have a better causal or like better generalization. And it turns out to be the case that when you have a better disentanglement, you actually have higher prediction power, as you can see it on the left. Yeah. So this can actually go beyond expression and omic data and could be applied to other type of like high throughput screening assays, which are actually cheaper. But actually they don't give you the gene expression or the phenotype, but they give you a morphology. And this is kind of basis of many drug discovery companies. And the most famous one is actually Recursion, that is based on this idea. And there are like assays to perform this type of like morphology-based screening. Like the most, I think, well-known one is this kind of, it's called cell painting. So this is actually used to screen many drugs and see how the morphology of the cell changes after a certain drug that could give you insight about the mode of action or mechanism of action of the drug. So the question is, can we train on this type of data set and then predict the effect of the morphology for unseen drug? This is very similar to type of like, you know, kind of style transfer idea, where you have a content image and a style image, and you want to kind of like, you know, combine a certain painting style with into an image. And yeah, so we kind of like took these ideas from counterfactual prediction and designed an architecture to actually encode the image at a single cell level and then predict the drug effect on the morphology. So yeah, I'll skip this part, but like what we observed was, if you give this model an unseen image, and so in this case, cytochalasin is a drug that actually triggers actin degradation. So those are like the red dots that you see in the prediction, and the model can rightly pick up these type of changes, even for unseen drugs and unseen perturbation on cells during the training. And the same thing for a drug that actually causes loss of integrity, nuclear integrity. But what people actually use in the domain is they don't use the image-based model. They actually use featurizers. So there are like softwares that actually take these images and then project them into well-defined like morphology-based features. So we took those generated images generated by this model, and we projected those into these like more interpretable feature space. And what we observed was indeed the prediction actually just selectively changes the actin structure as you see it in the image, but also based on these expert extracted features, you can see the baseline, which are the control cells, and the true drug, so the phenotype from the true drug, and then the prediction from our model that actually see a shift from the baseline toward the true phenotype. But what is interesting is if you can now use this model to actually predict the unseen drugs. So here we performed a kind of control experiments where we held out a bunch of drugs where they had similar chemical structure to the training, and also a bunch of them had kind of like dissimilar structure to the and that's what you see on this heat map, that certain drugs are very similar to the training. But in all those cases, no image from these drugs were seen, right? And at the test time, we want to see if the model could actually generalize to unseen perturbations. So I'll move to the benchmark that so this model is kind of like so we have this model there's only one work out there that actually tackled the task, but by the time that we were working on this, so this work still is in revision Nature Methods, but there seems to be a very high it's a very challenging task, and then what we show is like you can get like up to like 85% improvement over like another existing model, which has got a multiple image, which is a flow-based model for this task. And then now we're working on like the other type of generative models, so diffusion-based and etc., to further improve this. Okay, I don't think I have much time, so but like I would also like to talk about like the last part of the talk, which is about the tissue heterogeneity. So I would just give it 5 more minutes if that's okay, Andres. To briefly talk about this part too, which I think is quite interesting, is so with single cell genomics, before single cell genomics, actually, this is a concept that with bulk genomics, you didn't have the exact resolution, or you didn't know the individual cells in that tissue sample. So it's kind of like a milkshake. With single cell genomics, now you have the fruits, so you know which fruits are actually in there. But with spatial omics, you don't just know the fruits, but also like in this case, you actually know where they are in the tissue, so the location of each cell. But there are multiple challenges to analyze this data. It's not really straightforward to integrate a bunch of spatial data sets because they all come from different coordinate systems. So for example, like one is from cortex, the other one is from like, another region in the brain. So you can't just like align them. The second question is, can we identify niches? So these are like spatial domains where certain cells are localized and these cells are communicating to each other. And finally, going on a molecular level and explain what are the ligand receptors or communication patterns that are governing these spatial domains. So we came up with a method that we call niche campus. So again, it's very similar to the first model that I showed, XPmax. So it's kind of biologically informed model that we formulate this task as a graph integration. So we model each spatial data set as a graph, and we try to kind of align these graphs in a latent space. So adjacency matrix is you can infer it based on the tissue, and each node in that graph could have different features. So you can have genes, you can have peaks, so from chromatin. So you can also have co-founders that could be like, or like covariates, the patient or the age, sex, and the other type of technical variability. And then we encode those samples, those graphs, into a latent space that each component of this latent space is actually defined by certain cell-cell communication pattern. So you have a certain ligand and receptor, certain ligand receptor and downstream targets. You even have the metabolites or transcription factors regulating certain communication patterns. So, and this comes from domain knowledge. And similar to the previous work, you can actually learn novel gene programs too. So those are the spatially variable genes. And what we do is we actually go back to deep mechanism like underlying the cell-cell communication. Because if you look at the cell-cell communication, you can look at from the sender cell and the receiver cell. So it's very similar to communication problems. And if you look at it from the sender cell, you would like to be able to predict what happens in your neighborhood. So that's why we divided like this task into multiple reconstruction tasks. So you have a mask decoder that actually for each sender cell, you would like to predict the receptor on your neighborhood, but also the target genes on your neighborhood, in addition to predicting the location of those neighbor cells. So this is a collaboration with many partners and here in Sanger, but also later in these slides I'll show with also partners from Yale and Karolinska. In this example, we applied this model to integrate 3 different mouse embryos across different somite stages. So what you see here is like the histology colored by different cell type in this data set. And we applied niche campus. And what you see at the bottom is actually the latent representation for this data set across 3 different embryos. And the way to validate this is, for example, we annotated like the different structure in the mouse brain. So what you see is, for example, forebrain. Ideally, the model would integrate, on the last part, on the embryo 3, that circle should be a bit like to the right and bottom, to integrate these 3 forebrain and structure in the tissue across these 3 embryos at the same place. So that's what you see on the right. And the interpretability from this method could actually give you a mechanism to explain what are the cell-cell communication pathways regulating this niche. So this set of cells that are talking to each other. And it turns out you can sparsely explain this different spatial domain and niches by taking the top 2 gene programs that explain each spatial domain. And if you take those features and perform a very simple clustering on them, you can reconstruct the higher structure in the tissue. So if you look at brain, for example, like forebrain, hindbrain, midbrain, and floor plate, they are kind of like combined with spinal cord. These are like all the kind of like very similar niches. They group together, so you can go back to the the anatomy. And so I'll skip this part, but what is nice is then it kind of gives you the high resolution information about like how different cells are talking to each other. And we show in this example that we can find more like very granular structure in brain called floor plate, which could not be previously identified using just individual embryos and gene expression. But what is, I think, from my point of view is interesting is now you can put all of like as a kind of like end-to-end analysis into a graph explaining what are the mechanism and gene programs that each niche and the tissue uses to talk to other niches. And this could be basis for further downstream applications, for example, whether we can like inhibit certain signal or induce a certain signal using certain drugs and could be very important for drug discovery applications. And yeah, so I'll skip the benchmarks, but yeah, so you can actually extend this to multiple modalities. So not just gene expression, collaboration with Rockfang from Yale and then Castello Branco from Karolinska Institute, where they came up with a very nice technology that you can not just capture the location, but also gene expression of the cells, but also capture the chromatin accessibility. So that gives you the ability to now perform this analysis and talk about the regulation patterns and also the epigenetic underlying basis of cell-cell communication, whether there is a certain transcription factor that, if activated, can activate downstream ligand that could cause communication between that ligand and with cells with activated ligand and receptor and targets. So you can like have more mechanistic aspect. And what I think like here, what we also showed is actually the sum is better than the individual. So you can get more structure in the mouse brain looking at the data. So basically integrating across chromatin accessibility in the gene expression. So that was it. But we there are like many other works. So we also work on dynamics, so generative modeling in general to predict the cell fate, building hierarchies, and also in immunology, where we work on combination of gene expression and also like T cell receptors. And ultimately, the goal in our lab is to improve cell engineering. So combining all of these generative models with large scale perturbation experiments to perform AI-assisted cell engineering for cell therapy and drug discovery applications. And in Cambridge, we're also located in a very nice environment, and I'm just happy to collaborate. And so this was like work from like many people and like acknowledging funders. And if you have, happy to take questions, but also, there are positions available certain in different stages. So if this kind of interests you and it kind of excites you, please feel free to reach out. Thank you very much.
Thanks, Mo. That was absolutely terrific tour de force. Please post questions on the chat. We've got the first one from Chin Yuang. Do you want to read the question out? Do you mind?
Yeah, sure. If you have any, okay, so do we get?
Oh no, I thought Chin Yuang was gonna.
Okay, sorry.
Yeah. You there?
Sorry, Andres. I have to take care of my child, and I'm just watching at the moment. If you can handle the Q&A.
Yeah, sure. So the question is, can you explain a bit more about how you constructed the neural network architecture to reflect the gene program information, and how did you learn new gene programs?
Yeah, sure. So the way that we enforce, as I kind of like briefly mentioned, these type of like disentangle is we make the decoder linear. That allows us to actually wire the weights of the neural network. So we just, we force the model that for each latent dimension, the model is only allowed to reconstruct certain genes. So it actually discards the other variation from other genes, and those genes are actually part of a certain gene program. And so this is a hard mask, but we also applied a soft mask. That is where actually the challenge comes from, that you need to have a soft mask to actually remove and add genes. And for the other question, for learning new gene programs, we assume we kind of like enforce a information bottleneck, and we give a very small capacity to the model to learn new gene programs. And so if there is novel variation in the query data set, the model has to go for the strongest variation that is independent to the previous gene programs. So prioritizing the unseen structure, unseen expression pattern compared to the reference set. Hope that kind of explained, but that is well documented in the paper.
Yeah, thank you. That's very helpful.
Great. Thank you. Next question then. Would integrating new atlases under the SCArches framework change the embeddings of the existing previously integrated?
No. That's the whole point, right? So we wanted to keep the map fixed, right? So you have a map, you don't want to change the coordinate because that kind of destroys the interpretability.
Great. Thank you. Maybe I can ask a quick question. So you briefly touched on it. I guess I'm kind of intrigued by the idea of moving in both directions across from one omics data set to another one. And you kind of mentioned the idea of tissue engineering or cellular engineering. So not only can you identify which cell programs are activated based on the expression data, but conversely, can you predict what would happen to the cell maps given a particular perturbation in a in a system. Do you want to say a bit more about the challenges?
Yeah, sure. So I think that the idea of cell engineering, what I kind of refer to, is like a bit different from like synthetic biology perspective. So we're basically talking about perturbation-based cell engineering. And in that case, we are trying to design. So in the lab, we have both like, data generation and also modeling. So in the data generation part, we kind of try to do, as I explained, with like sparse perturbation experiments. And then if we have a certain phenotype, for example, like in one study, we are trying to, in one like ongoing research in the lab, we study neuroinflammation and how you can modulate neuroinflammation. And then you have certain GWAS and like candidate genes that could either modulate or induce or inhibit certain inflammation pattern. So the question is, if we want to modulate that, what are the perturbations? So you need to combine this sparse experiment with a generative AI that could generate cells for unseen experiments. And there is a big challenge here because we are doing all of these stuff in vitro and organoid models. So now the question is how to put it back into in vivo. So that's why the whole part about like in vivo to in vitro mapping comes to play. And it's kind of like, all feeding each other. Yeah.
Super cool. Super cool. Maybe, well, Mo, can I just ask, have you looked at kind of diagnostic potential for your models? That's to say, can you, given a certain single cell RNA-seq data, can you predict something about the state of the individual in terms of, I mean, is that how easy?
Yeah, so that's basically where we're going, with this type of like multi-instance learning stuff. So there we actually take a lot of like patient data, so not just like omic data, but also like EHR information. And then we're interested to like kind of like more toward like personalized medicine, predict which cells in which patient actually changes, and then correlate that with the metadata. And that if like, ideally, that could inform us about the type of treatments would have for each.
Yeah. Super cool. Super cool. Mo, thank you again.
Thank you very much for having me.
Thanks to everyone. And great.
Nice
okay, fabulous.
Bye.
Okay, thanks.